We're about to get started. If you don't mind settling in. So the session we're about to do here, I'm very excited about. It's going to be, we're going to hear from several people that do some really innovative, cool things with tissue imaging using the Novo Mass Spectrometry and MALDI. And so, so the big difference here compared with something like, IMC or MIBI is that this is done where essentially the mass spec is done so that you're analyzing directly sort of endogenous native chemical species from the tissue. So it doesn't require the use of labels or anything like that. And so it opens up sort of a whole world of really complex, biomolecules that you can analyze directly that, really aren't tractable to look at with target approaches like, like glycans and lipids and stuff in ECM and everything. So to kick us off with that, Jeff Spraggins is going to talk to us about some of the stuff that he's been doing, I think, with the kidney atlas and also just in general, the hopefully a magical mystery tour of his MALDI stuff.
Thanks a lot, Mike. Yeah, so my name is Jeff Spraggins. I'm an associate professor in cell and developmental biology at Vanderbilt University. And today I'm going to talk about imaging mass spectrometry, as Mike said, and how we think about integrating imaging mass spec with other forms of molecular imaging. My research group, I think about our research program in kind of two pieces. First is developing the technologies focused on imaging mass spec, so all the similar things that everybody else works on, sensitivity, specificity, spatial resolution, and improving those aspects of the technology. But I think what our group is kind of known for is strategies for combining imaging mass spec with microscopy and spatial transcriptomics and other things, both the experimental workflows that enable that, as well as the computational tools to allow us to mine these data. Imaging mass spec, I think, is a really unique form of molecular imaging to serve this purpose, this kind of glue modality between the various forms of biomedical imaging. And that's because it's untargeted, as Mike pointed out. So we're looking at the endogenous molecules within the tissue. It's highly multiplexed. So typically we're looking at hundreds or sometimes thousands of biomolecules simultaneously. And we can apply it to really any molecular class that we can detect with a mass spectrometer. So we can look at intact proteins, peptides, glycans, which you'll see in the next talks, lipids and metabolites, which you'll see here. And it's this combination of high spatial fidelity or spatial information combined with the deep molecular coverage that allows it to act as this bridging modality with things like tagged microscopy on one side, as well as spatial transcriptomics and kind of classical multiomics on the other. Now, since I'm the first talk in this session, I do want to do a little bit of teaching. There are various kinds of imaging mass spectrometry. Our version is what's called matrix-assisted laser desorption ionization. I think this is the technology that everybody speaking today will be covering. This is a laser-based approach. This setup is very similar to a standard microscopy experiment. So we start off with thin tissue sections, usually 5 to 10 microns in thickness. We tend to work with fresh frozen samples, so they're cryosection. Although you can work with fixed samples, I think you'll see examples of that later as well. Once the tissue is mounted onto the slide, we apply a chemical matrix. This matrix is there, it's chosen because it's going to absorb the wavelength of our laser very efficiently. And that molecule is going to help with desorption and ionization of the endogenous molecules. And then we simply go from pixel to pixel, firing our laser, generating ions and detecting these ions within the mass spectrometer. So you can think of every pixel in our experiment as a separate mass spec experiment. So what we end up with are hundreds of thousands or millions of mass spectrum, one for each pixel, and we can represent the intensity of any of the ions we detect as a heat map, or what we call an ion image, and that's how the experiment's done. Now, just to give a quick schematic, so this is a pretty common mass spectrometer. This was developed in our lab in collaboration with Bruker Daltonics a few years back. This is now referred to as the MALDI timsTOF Flex. This was kind of the first prototype system, which was just a modified Pro. And effectively what we have is the laser system. This is a neodymium YAG laser. It's a 10 kilohertz laser. We fire the laser and focus it onto the surface. The ions then pass through this funnel, which does exactly what it sounds like. It funnels the ions into the mass spectrometer. We can do some cool things up front here. We can separate ions based on their gas phase characteristics, but I'm not going to talk about that today. They pass through a set of multipoles. which allow us to isolate and fragment ions to get more chemical specificity. And then they enter an orthogonal reflectron time of flight. This is just a really nice mass spectrometer for imaging because it's very fast and it gives us very good spectral characteristics. So good mass accuracy and good resolving power. What it really boils down to is with this particular setup, we can achieve pixel sizes of about 5 microns. So that's dependent on how tightly we can focus the laser onto the surface. And we can do it relatively quickly. So we can generate images at about 30 pixels per second. So we're generating 30 mass spec experiments per second. So an example. So this is some work. All the data you're going to see today is part of the Human Biomolecular Atlas program, or HubMap. This is our work with the human eye. And so this data set is from a 37-year-old donor. This is the retina of the human eye. And it's collected on this timsTOF Pro in positive ion mode. You can run it in any in either polarity to look at different kinds of molecules. And this is a 5 micron imaging data set. So we're looking at lipids and metabolites here from mass to charge 500 to 1,000. Although we collect a little wider than that, I'm just zoomed in. And just to zoom in here, just to show how complex these spectra are. So even at 5 micron resolution, we're detecting thousands of molecular species. Any of those we can represent it as ion images. So as I said, this is human retina. And the retina is a nice kind of starting point example of why you need this kind of resolution, because you have this very fine structure in the human retina. So we have these layers. So we're looking at an H&E collected after the fact here. And I just want to point out this one layer that's darker in the H&E. This is the retinal pigment epithelial layer. This is a single cell layer within the retina that feeds and helps kind of cycle metabolites within the photoreceptors. So in this overlay image here, which is an overlay of 4 ions, these are all false color scaled. So we're just picking a color to represent as the intensity. This ion in red at mass to charge 592. Below here are the individual ion images. So we're looking at the one on the left. This is a molecule A2E. This is a known retinoid metabolite. This is localizing to this single cell layer within the retina. So this just shows that although this is not a single cell imaging technology, I wouldn't call it that, it's certainly approaching cellular resolution. So we can link untargeted molecular profiles to specific cell types within the tissue microenvironment. Of course, we can do this for any tissue type. So this is an example of some of our data from the human kidney. And everything you'll see after this is associated with our kidney studies. And what you're looking at here, these are a little bit tricky to look at because these samples are so large. But the top right corner is the medulla. This is an overlay of these 6 molecules here, and their associated colors are listed in the same order below. And so we have the medulla, and the primary portion of this image is all the cortex. And so just to orient you a little bit, you probably all know this, but on the left here is just some schematics of the anatomy of the kidney. So we have the cortex and medulla. In the cortex of the kidney, we have millions of nephrons, and that's what we're representing here in this biorender schematic. And so in our image on the right, in light blue, these are the glomeruli, these are the filtration units. And then we've selected different ion images because these particular lipids localize to specific tubules. So the proximal tubule, the descending and ascending limb of the loop of Henle, the distal tubule, and the collecting duct. And if you freeze your tissue well enough, this is all fresh frozen tissue, you can even make out the lumen. So if you're very careful about how you prepare these samples, you don't collapse the tubules. You can still make out a lot of the tissue morphology in these images. Okay, so I just bragged about what we can do with imaging mass spec, and I want to talk about what's difficult about the experiment. So we're quite good on the molecular side of things, right? We're using the mass spectrometer, so we're highly specific. As I said, it's highly multiplexed. But on the spatial side, it's a little tricky, right? The imaging type is odd. We have relatively low spatial resolution relative to microscopy, so we have fewer pixels. But our information density is very high. So the raw data coming off these platforms can be millions of bins. So you have lots of channels in a single pixel. Usually, as I said, it distills down to hundreds or maybe a couple thousand molecules. And importantly, for a lot of the molecules that we're looking at, lipids, metabolites, glycans, there may not be well-established set of literature saying, hey, this lipid or metabolite or this profile is associated with this cell type. That's just not available, right? So we don't have that direct connection. So this is where imaging mass spec is weak. And so our approach to deal with that is through multimodal analysis. And so we combine imaging mass spec, in this case, in the example I'm going to show today, with microscopy To give that biological context, we're going to first co-register the imaging mass spec data with high accuracy and precision to the microscopy. And this will allow us to then do some really unique forms of multimodal data mining, which you'll see, with the goal of connecting each of our mass spec pixels specifically to functional tissue units, cell types, and eventually single cells. So just an example of a multimodal data set. This is a data set from a 38-year-old female donor. This is an autofluorescence image. This is a whole slide image. So all the microscopy data we collect are always whole slide images. Although on the next slide, you're going to see data from just this square. This is where we collected the imaging mass spectrometry data from. And in this autofluorescence image, you can clearly make out the cortex and the medulla. And so in the next set of images, the bottom will be the medulla and the top will be the cortex. And this is what the data look like. Okay, so left of the white line, everything left of the white line is collected on the same piece of tissue. So we start off with autofluorescence microscopy. We generate the imaging mass spec data set. These are just 3 example lipids, a sphingomyelin and 2 phosphatidylcholines and their distributions. But again, we have thousands of molecular distributions. And importantly, we don't damage the tissue. If you run the experiment appropriately, you're just etching off that matrix. You're not actually damaging the tissue. So we stain the tissue afterwards, in this case with a PAS, which is a standard stain for a kidney pathologist. To the right of the white line, we collect autofluorescence. I'm not showing that because it's redundant, as well as multiplex immunofluorescence. And each of these modalities have a specific purpose. The autofluorescence we use specifically for co-registration. I don't have time to get into that today, but it's our common modality that we use to link all of our data sets together. And we use it for segmentation. I'll talk about some of those results. Obviously, the imaging mass spec gives us untargeted molecular profiles. PAS gives us a direct link to classical pathology. And then I'll talk about what we do with multiplex immunofluorescence to help inform some of the segmentation maps. Okay, so in terms of segmentation, we are doing things at slightly different scales. So you learned a lot about segmentation yesterday, particularly at the single cell level. We're really more interested in kind of the FTU level, the multicellular functional tissue unit level. So segmenting out all the components of the nephron in this case. But the trick is we don't want to do it on multiplex immunofluorescence. We want to do it on autofluorescence because we can collect that everywhere and we get it for free. So what we're going to do is develop an autofluorescence-based functional tissue unit segmentation pipeline. We're going to use the multiplex immunofluorescence to guide that process, to give us lots of examples to teach the computer to build robust models. And ultimately, we're going to use a CNN to do this. And so this is what the data look like that we use to build these models. And I'm not going to spend a lot of time on this, but effectively, we can collect the autofluorescence first. I should point out this is 3 channels, so red, green, blue autofluorescence. And then we collect the multiplex immunofluorescence. Of course, we're choosing markers that are marking the specific functional tissue units of interest. The multiplex immunofluorescence, as all of us are aware, is very specific. So it doesn't take many examples to train a computer to recognize functional tissue units based on multiplex immunofluorescence. So we can go from very few examples, train a computer to mark many tens to hundreds of thousands of examples of functional tissue units across many patient samples. We curate those, and once we have all of those examples, we then transfer them to the autofluorescence where we need more examples to build robust models. And this is what we get. So when we feed now our algorithms with autofluorescence data, we get full segmentation maps of the functional tissue units. Again, not single cell level segmentation, but multicellular functional tissue units within the kidney. But for our work, this is appropriate. So what do we do with that? So the easiest thing we can do is all these data are spatially co-registered. So on the top row here, you're looking at the imaging mass spectrometry data, and they're just 4 selected ions. Below that are those same images now overlaid with the segmentation maps. And we've just simply used a Pearson's correlation to figure out which molecular ions are most closely correlated to the segmentation map. And so 2 examples to highlight. On the left is a phosphatidylethanolamin. This is an ether lipid. That's the most highly correlated to the segmentation map of the collecting ducts. And probably the most striking example is this phosphatidylserine 44,4 on the right here. That's the most closely associated ion or correlated ion to the segmentation map of the thick ascending limb. So this just gives us a way to kind of filter down the thousands of ions that we detect down to those that are most highly correlated to anything that we have a segmentation mask for. This works great. For a single example, but it doesn't work so well if you're trying to do this across large patient cohorts and find larger trends in larger cohorts of data. And so the way we deal with that problem is we build classification models. And so this is done in collaboration with Rob Vanderbilt's lab and his graduate student, Leonard Tideman. And so this is pretty standard. The way people build classification models with mass spec data is you're basically going to feed in mass spectra. In our case, these are associated with pixels. And then you'll have your favorite researcher come and label those pixels, and you'll build a classification model. Of course, we don't want to do that. We're going to use the segmentation masks that we've automatically generated with the autofluorescence. So we can do this across all of our patients and across hundreds of thousands of instances of functional tissue units. So we can build very robust classification models. And then normally what people do is they show up with new spectra and they say, okay, computer, tell us what functional tissue units that's associated with. We don't really care about that in this case, right? We already know how to segment the data. We can already associate those pixels with the segmentation map. So what we want to do instead is we want to open the model up and figure out how it made its choice. Because if we can figure out how the models are making their decisions, what molecules are driving those decisions, it gives us the ability to do in situ, spatially driven biomarker discovery. And so we're using interpretable machine learning to open up each of the classification models using Shapley additive explanations. This gives us a ranked list of biomarker candidates for each functional tissue unit that we have a segmentation mask for, and ultimately allows us to chew on data that looks like this. So this is a mosaic of mosaics. Each of these squares in this image that you make out is imaging data from a different donor. Okay, and then within that square are all the little thumbnails of some of the molecular images, and it's only a portion of the data. So in this data set, we have 33 patient samples. We run it in both positive and negative modes, so we actually have double that in terms of the imaging mass spec data. And that leads to about 30 million mass spec experiments, and again, tens to hundreds of thousands of instances of functional tissue units. And we can take all this data with this pipeline and distill it down into a ranked list of the most important markers, in this case for the glomerulus. So what you're looking at here is the global SHAP value for the top ions that are associated with the classification model for predicting glomeruli across all of these data. And it's color-coded. So the higher the SHAP value, the more important that is as a molecular marker. And it's color-coded based on whether or not it's positively or negatively correlated, right? Because it's not necessarily just that those molecules are present that define a glomerulus. It could be that those molecules are absent. This is one way to look at the data. I kind of like this better, although it's a little bit intense. Let me walk you through it. What we have along the top are different lipids and metabolites, and then they're grouped. We've just sub-selected the data, so it's about 10 different donors. It's the same donors in each case down here, and the data are just grouped based on functional tissue unit, glomeruli, proximal tubules, distal tubules, and so forth. The size of the dot represents the global SHAP value. So that gives you a sense of how important that molecule is as a marker. And then again, the color is linked to whether it's positively or negatively correlated. And the thing I like about this is right away, you can pick out all the molecules that are kind of rock solid molecular markers across all the patients. So just to zoom in on a couple examples for the glomerulus and the proximal tubule. For instance, the sphingomyelin here is one of the most important molecular markers of the glomerulus across all of our patient samples. For the proximal tubule, same can be said for this phosphatidylethanolamine and this phosphatidylserine. But this phosphatidylserine is also a marker for the glomeruli, however, much more highly variable. And what we find is that in some of these molecules, of course, they're linked to other comorbidities that these patients have: age, sex, obesity, things like that. All right, I wish I could tell you more about what we do, but I think I'm probably already over time. What I hope to show you today is some of our workflows for integrating imaging mass spectrometry, this untargeted, unique form of molecular imaging with other types of modalities, and some of the tools that we're using to integrate and mine the data, and ultimately how we're using both machine learning and interpretable machine learning to deal with mining across these large data sets. So with that, lots of people to thank. My research group here, this is a lovely lunch we had this summer. The rest of our HubMap team that are associated with our U54s, our collaborators, and the rest of the Mass Spec Research Center. But with that, thank you very much.
Hi. Thank you very much for the talk. It's been very interesting. Would MALDI be able to resolve isomers in situ?
Yeah. Yeah, so the question is whether or not we can resolve isomers. So you can do it two ways. If you're just based on intact mass, no, of course they'll have the same mass. But we have a couple of papers and others have done this where I mentioned what you can do in the front end of that instrument in the funnel. You can separate things in the gas phase based on their three-dimensional conformation. And some isomers we can distinguish based on that using what's called collisional cross-section of the molecule. The other way we can do it is by doing pixel-wise fragmentation of the molecules to differentiate the isomers.
Okay, and the next question would be, would you use MALDI for, so if you have to choose a molecule, let's say lipids, metabolites, RNA proteins, which would you use to use MALDI for?
So lipids and metabolites, kind of for sure. There's not a lot else that you could use that will provide that level of specificity, not necessarily MALDI, but imaging mass spectrometry and the other, because there's other kinds of sources and ways of sampling. For proteins, it can be a little bit tricky. So I didn't get into it today, but for proteins, we are limited in mass range. So with MALDI, we're generally dealing with low charge states, which for all the mass spectrometrists in the room, we can appreciate this, but it's really hard to move a very large ion with very few charges through a mass spectrometer. So we tend to be limited to sub 60 kilodalton proteins. So we're limited to smaller proteins, and then the identification of those proteins can be tricky in some cases. So if you have a very targeted protein that you have an antibody for, go do immunofluorescence. That's what I would tell our collaborators. If you have some unknown thing that you're looking for, then maybe imaging mass spec is the right tool.
Thank you so much.
Mhm.
Yeah, very impressive. I was wondering, coming from a regular mass spec to this kind of mass spec, whether you had to make a new library in order to identify each of your metabolites and lipids, and whether the retention times and the masses change when you do this compared to just putting a sample in a MSMS. Or...
Yeah. So the masses don't change, right? The mass is the mass. So that part doesn't change. We don't have a retention time. So we're not using retention time as a way to align those two data sets. You can use just a publicly available database to get kind of tentative identifications. So lipid maps, Metlin, those kinds of things you can throw your data against to get tentative IDs. And then what we tend to do is for all of our publishable data, we'll run LCMS to get kind of a custom database that we compare all of our experiments to. And if there's still any questions about an identity, then we'll do fragmentation directly from tissue, and that answers it.
Thank you.
Yep.
Hi, that's a really cool talk. So coming from an immunofluorescence background, I know you mentioned there's like this sort of 60 kilodalton, maybe boundary of identifying proteins. So I'm kind of curious. So in the single pixel where you get this spectrum, like how convolved is that? Like how many proteins do you think go into that? And like how many can you reasonably identify? Do you use nearby pixels to kind of, improve your fit and identification?
Yeah, it's not that, so yeah, we don't need to necessarily deconvolve on the spectral side. It's not like a single spectrum has a different resolving power or the narrowness of the peak than the next or an average. What can happen sometimes if you have misalignment is if you're only looking at the average spectrum, your peaks can broaden a little bit. But we do all sorts of pre-processing to make sure everything's aligned so we don't have that problem. What you can have are sensitivity issues. So from a single pixel, you might not have a very good looking spectrum, but over the course of a million pixels, it averages out to have reasonable signal to noise. So we don't really have that issue of having to kind of deconvolve things in that way. What I will say is it depends on the instrument how many proteins you detect. So if you're using a linear time of flight system, which is more traditional for proteins, tens of proteins. If you're using an FTICR or a QTOF like this, now you're talking about hundreds of proteins. So it just depends on how much resolving power and performance you have.
Thank you.
A couple questions, and then we'll get on to Peggy from the online Q&A. There are several in here that are essentially asking about like sample preservation compatibility. Can you do this on, what can you do on FFPE? And then along those lines, I was just going to ask one of the things that I've had people ask about a lot is using MALDI for metabolite imaging. And I've always been really freaked out to try that because I've always gotten the impression that the sample prep is like, can be difficult. So I think there might be a lot of people that'd be curious about that in terms of the
Sure.
complexity of that. What do you think about metabolite imaging?
Yeah, there's two questions there. So first, I think you're probably going to hear FFP experiments. So that's totally doable. If you're adding enzymes, particularly in the N-glycan experiments, or if you're looking at, tryptic digest of proteins, which you can do, you can spray trypsin to get at larger proteins. FFP is fine. We actually get reasonably good lipid data from FFP samples. That's not so much of an issue. The fixation is not the problem, it's the paraffin. So it's just, can you trim away enough paraffin and then use a couple of washes to get rid of it without distorting the molecular distributions in the tissue? Because the paraffin just ionizes like crazy. And so it's all a competition for charge. So if that paraffin's around, it's going to suck up all your charge and your sensitivity is going to be terrible for the endogenous molecules. For metabolites, it is harder than lipids for sure. It's for a couple of reasons. One, ionization efficiency for certain metabolites is going to be lower than lipids. So again, it's a competition for charge. And then two, the mass spectrometers tend to have been built for proteomics, which means they're really, really sensitive from like mass to charge 400 to 2000. And if you go outside of that mass range, now you got to do some interesting things to tune them to make them work well. So if you're dealing with smaller metabolites, then you have to make sure you're tuned or get new power supplies or something to help you with sensitivity in the low mass range. The other thing to think about with metabolites is you can have issues if you're dealing with fresh frozen samples, where if you're not really careful about warm ischemia or something like that, you can have real issues with distorting the metabolite profiles very quickly in those tissues. So you have to work really hard to freeze them quickly. And then we try not to store. If we're going to do metabolite imaging, we tend to freeze and then run within the week. We don't like to have those stored for long periods of time.
Thank you.
Yeah.
So next up, we've got Peggi Angel from MUSC, who is going to talk to us about some of the great stuff that she's doing with MALDI to explore the extracellular matrix.
Thanks, Mike. I don't know if I should give I think everybody can hear me just fine. And so like Mike was saying, I'm going to be talking about the matrix. And so a lot of times we've been talking about cells. These are my disclosures. And so when we think about single cells evolving and progressing into disease, we need to think about how they communicate. How do they recruit immune cells to their area? How do they establish these long distance and short distance signaling perimeters? Well, they do this through the extracellular environment. And so this is called the matrix. And this is a three-dimensional space that is full of chemistry, chemical biology, and structure, and flux. And so as our biology changes, this extracellular environment changes as well. And so what I'm going to talk about today is the encoded collagen structure. And so collagens are main structural features of the tissue. And so the triple helical region specifically, let's see if I can get this to work, and I cannot. Let's see if I can get this to work. So what I'm going to be talking about specifically is this triple helical structure and how it varies in disease states and health. And so what you'll see here is an image of these different variations in triple helical structure in hepatocellular carcinoma. And so the point to get across is that the THR is an encoded structure that varies spatially and with cellularity in disease, and it has a very high predictive value. Okay. I'm learning the system here. It's not progressing. Thank you. Okay, I appreciate that. Thank you. We'll go back a few slides. Okay, and so I'm going to start by using as an example collagen alpha 1,1. And so this is a protein that people think about as a singular protein, but it's actually very complex in structure. It's complex in turnover and organization. And so if we look at the pictures here, we see that collagen fibrils in skin have a crisscross pattern. This gives our skin our stretchiness, our function. And here we see collagen fibrils in cornea, which push out the cornea, giving us the ability to see at a certain resolution. And so although this same protein is present in every single tissue, how it's organized has different function. This organization is driven by a post-translational modification called hydroxylated proline. And what this hydroxylated proline does is it forms hydrogen bonds between these triple helical structures that give it its binding pattern. And so the opening and closing of this triple helical structure is what allows proteins like integrins and other different receptors to bind to the structure at different levels. And so here I'll show a part of the protein that will be seen later where we have hydroxylated proline has high affinity binding. And so these structures are actually prepared in the cell is secreted outside of the cell as this triple helical structure. So outside of the cell is where they are assembled into collagen fibrils and collagen fibrils, which give it the entire composition of the tissue. Now packed within this triple helical structure are encoded these different domains that cause different biologies to happen. So if we unravel one of those structures, we will see these different domains that interact that cause angiogenesis, that bind to endocrine receptors, that change the organization and composition of the tissue. And so what we know is that if we look at cancer in particular, we know that this collagen structure has predictive value. The problem is that these values that have been so forth read are things that are very qualitative. They're alignment, collagen type, number of collagen fibers, and post-translational modification. But this is given by transcriptomics, by the enzymes that place these modifications on the collagen structure. And so what we need to get to is the exact problem of how can we get to the molecular composition of this structure. So again, going back to collagen alpha 1,1, we can look at its structure and how it's formed singularly. And we see that within the triple helical region, there's 235 prolines that can or cannot be modified. And so this is what's really changing the composition of the extracellular environment. And then we have some other parts, and we're going to look at other post-translational modifications later. And so how do we get to these huge proteins that have tertiary and other superstructures? Well, we use enzymes, not antibodies. And in this case, what we're doing is we've developed a strategy using collagenase type 3. And so this particular enzyme binds to the triple helical structure and then crawls along it, chewing it into strands. And these strands are what we're detecting by mass spectrometry. And importantly, this is applicable to clinically archived tissues done without decellularization. So we can perform these on any tissues that you can pull off of your shelf. And I'm going to show you also that you can do this on any of the tissues that have already been stained. And so we not only pull down this collagenase, with collagenase, these collagen types, but we pull down around 40 to 60 other ECM proteins that are involved in collagen regulation. And so I'm not going to go into depth through this because Jeff did such a great job, but we start with patient consenting. Pathologist annotates the H&E slide. We do an immunohistochemistry like pre-preparation. This is on FFPE. It also works on frozen. And then we use enzymes, not antibodies, to access these structures. And then we look at these little pieces of domains, which we call peptides, that were detected by imaging mass spectrometry. So importantly, it's integrated with common pathology workflows. It uses standard microscope slides. There's no extra sample collection from the patient. And so this is an example of what we might get. This is from hepatocellular carcinoma. You can see that there's a high complexity of spectra, and there's a huge amount of spatial data that we get. And we can do heuristic spatial clustering. And what we're seeing here with bisecting k-means and Manhattan metric is that we get a segmentation of the extracellular matrix. This one is rather simple, but every single color here that is shown is a different extracellular proteome. Now we take these tissues and we scrape them off of the slide, and we get patient-specific proteomics. And so from this is an example from this exact tissue where we get many different types of collagen, but also different types of extracellular proteins that modify those collagen structures. Now this is different from decellularization, because in decellularization, some of these smaller proteins are going to be lost. So you're disrupting this composition of what forms these collagen proteins. And so with this enzyme, our main spectral sampling is actually with fibrillar collagens. And then our other extracellular proteins represent around 20 to 9% of the sampling. And so when we look at the matrix proteome, and we look at this list of proteins, we can go in and do an in silico analysis and say what type of post-translational modifications do we see. So for collagens, it's a lot of hydroxylated proline, and this is what we're interested in. For the other ECM proteins that guide those collagens into place, we have phosphorylation and N-link glycosylation. And Rick is going to talk about N-link glycosylation in the next section. And our group has actually worked very collaboratively to produce not only just mechanisms using the concept of enzymes, not antibodies, focused on glycosylation patterns, to produce approaches that allow us to go from tissue to cells to biofluids. So we can go back and forth, we can go very translational, we can actually, if we wanted to, and if we had the right collaboration, we could collect some serum, for instance, from a patient at the bedside and analyze that for monitoring. And so going back to this concept to access the matrix through enzyme product imaging, we're using PNGSF prime, collagenase type 3, elastase. We have some other enzymes that are also in the works as well. And importantly, this is unbiased to species and organ type. So this is an example of integration of H&E pathology staining and glycan imaging in ECM. And what we found is if we remove these large sterically hindered structures, we get a pretty large increase in detection of the extracellular microenvironment. Importantly, since we're doing the H&E first, we can do second generation harmonics and then remove the cover slip and do deglycosylation. So in this case, what we did is we did multiple enzymes. We did cyclic application of enzymes followed by imaging. And so we did this over 7 different enzymes. And here we are targeting components of the extracellular matrix that included chondroitins, N-link glycans, elastins, collagenase. We did trypsin as well, not reported in that particular article. And so another student of mine, Denise Rashanarang, who's now at Cedars-Sinai, she demonstrated that you could do integrated immunohistochemistry and ECM targeted imaging proteomics. And this is really important because you can stain either in a singleplex or multiplex fashion for a particular enzyme and get that pattern across the tissue. But even if you see that, it doesn't tell you where the modification has occurred. And that's what MALDI imaging mass spectrometry is doing. It's telling you that this proline has a high chance of modification. This proline is modified, this proline is not modified. So we start to get this code of where the collagen structure is modified. And I want to also point out that if we look at the transcriptomics data, we can see this enzyme is upregulated as well, but again, it doesn't tell us where in the structure. So we've recently submitted an article looking at combining antibody stains with N-glycan and collagen imaging. This was led by Jacqueline Dunn, a student in my lab. And so she has used the Ambergen MALDI IHC capabilities, which basically attaches as a secondary photocleavable mass tag to the antibodies. And so our finding was that we could, using different approaches that use antibodies, we found that if we did antibodies first, followed by the N-glycan and collagen, then we got the best response from all modalities. And so we can certainly do a lot of multiplexed immunohistochemistry type of approaches. So now I'm going to talk very briefly about ductal carcinoma in situ. This is a project being led by a very talented MD PhD student, Taylor Houlihan. So we've heard about it a little bit yesterday. So ductal carcinoma in situ is a benign breast disease. It's diagnosed in around 25% of women, but which patients progress to invasive breast cancer is unknown. And most women opt for anything from lumpectomies to full mastectomies in order to just get rid of the worry about it progressing. And so I want to also mention that this is a extremely talented team that we are working with that includes people from Duke, Shelley Huang, Jeffrey Marks, people from Stanford, Rob West, Mike Angelo. We have another pathologist at MUSC, Rick Drake's group's working on the glycans, and Elizabeth Wallace, his student. And so when we just look at some of the images here, this is ductal carcinoma in situ grade 3. And so we can see as we go across the tissue toward the cancer region, we have this type of gradient pattern. And we can see these gradient distributions of different parts of the extracellular matrix. We do get some cellular proteins, which are very important to see. And so here we're seeing a piece of filament C that promotes cancer development. And then here we see an integrin binding motif across the tissue. And so in this particular tissue, there's no ductal carcinoma in situ, and instead it's invasive ductal carcinoma. And so here's where the tumor is located. And again, we can see these gradients across the tissue and see that this particular collagen that was presented as a gradient low to high in ductal carcinoma in situ is localized in the tumor. So Taylor has done a lot of work. She's getting very close to submitting a manuscript, but this is one example where we're showing the integrin binding motif and its variation as distance from tumor, as seen over here. Here's the tumor, and here's a lesion number 40. And then we see this variation with a part of collagen 3A1. And one of my collaborators, Javier Bravo Cordero, just published a paper last year talking about how collagen 3A1 limits tumor metastasis. So what we're seeing in this case is that we have this increase in this particular segment of collagen 3, increasing with distance from the tumor and decreasing around the tumor lesions. So that's some of the interesting information. But what Taylor has done really is gone through, and she's looked at the sensitivity and specificity of distinguishing between IDC and DCIS, and she's found a number of different peptides there. She's also taken knowledge and literature of elastin in breast fibroadenomas and in breast tumors, and she's used elastase to look at the structure of elastin. And it turns out that elastin is really specific about determining the different types of lesions. So this is an example of a combined image from that in ductal carcinoma. And this is actually the segmented image where you see lesions outside that are pink and lesions within the tumor are orange. And so what we've done is we work with Yoon-hee Park at University of Wisconsin-Madison, and she's done some exploratory work looking at area under the receiver operating curve and distinguishing between DCIS and IBC. And so we have some really good sensitivities here. And this is kind of characteristic of what we're seeing. And so in my last minute, I'm just going to romp you through a couple other examples. And so this is reactive stroma in prostate cancer. And so what you can see here is that as the modification changes, the localization intensity changes. So we're changing the actual landscape of the tissue as we develop these diseases. So this is one by Alex. And so here we're looking at colorectal cancer. Again, we have very high predictive value between stage 2+3 and 3+4. So we're looking for more, a larger cohort to test that on. This is work with Dr. Oscar Suhun's lab at MUSC. So here we're using patient-derived tumor cells and doing mouse implants and doing drug treatment, and you can see the therapeutic response there. This works very well in brain. So here we're showing just differentiation in normal gray matter and blood vessels. And again, these prolines are the ones that are modified. In this case, they're completely modified, but they have a different distribution in brain. And then last, in the Bay Area here, a group that's working on bone. And so they're looking at control versus osteoarthritis, and they're looking at the changes in the cartilage, which are something that cannot be detected by an X-ray. And so what we're seeing here is that there's very distinctive changes within the area that you have osteoarthritis, and they're actually developing some very predictive signatures of this. So with that, I'll conclude, and I just want to say that the extracellular microenvironment, I believe, is the next spatial frontier. It does control these gradients. It does control these potentials. It forms these hills and mountains that Russ was talking about. There's a largely undefined relationship with the cellular populations, and so we need to connect the landscape with the cells and understand why the cells have moved into certain areas. Post-translational modification is critical to this. So this is really what is controlling the gradient of these hydrophilic or hydrophobic molecules across the tissue. And then multiplexed enzyme imaging seems to be a very useful way to survey these different patterns in these large protein structures. And then last, the matrix has predictive value. and I couldn't go into it as much. So I want to acknowledge the patients and donors. I very much appreciate all of the collaborators. I am seeking an ambitious postdoc who will look at the brain ECM and Alzheimer's disease. And so we have some new ways. So if someone wants to step into that and write, that will probably make their career. And so then last, I want to advertise that we do have an imaging mass spectrometry conference coming up in Montreal. So this will be a worldwide gathering of people doing mass spectrometry imaging. We do a lot of integrative workflows and work with AI, and everybody is welcome to attend this. So thank you.
That was great. We have some questions from online. Enzyme imaging is fascinating, and I am new to this. Could you point me to where beginners could start with?
Well, there's several hands-on workshops. I would highly recommend the workshop that is held at MUSC. We have 9, we only allow 9 people to attend. And so we give very supervised care. We have developed protocols for looking at glycans. So glycans are a great interaction between the proteins and the metabolites. And so I would say that that's a great place to start.
And so I've got a question. So
Yeah.
So what's your record at this point for enzymes on a single slide?
Yeah, it kind of,
You can brag.
some of
It's
the
okay.
tissue. You know, the other thing we could do is integrate lipids and enzyme imaging, and that's something that I think Aaron Seeley is working on. But I think right now is about 9, but it's really limited by how patient we are and trying to understand, you know, because we have to remove the tissue to see if we want to get a really good picture of the sequences that we're looking at.
And then some questions from the online chat. People are asking, what's your antigen retrieval strategy? Yeah,
So in antibody imaging, you antigen retrieve to expose an epitope for binding, right? It's similar here, except here you're exposing an epitope for protease cleavage, right? And so you do have to go through the initial test to see what antigen retrieval exposes the right epitope for protease cleavage.
Thank you for your talk.
Mhm.
I was wondering, you mentioned how there's the associations between ECM density or different kind of characteristics with cancer outcomes.
Mhm.
Are you able to link these changes in ECM post-translation modifications with mechanical properties or any of those
Yeah,
features
so
of the breast?
that's my background, but I haven't done that yet. I would look for a collaborator that would want to do that.
Thank you.
So yeah, that's definitely a way to go.
I just have a quick question. So
Yeah.
when you find this beautiful work as an
Sure.
example, the different signals between the DCIS and the IBC,
Mhm.
how long to get those sorts of things into sort of a clinical setting where somebody can do a biopsy, test it and say, oh, you know, we don't have to tell this, we'll tell this person not to worry. I mean, does the NCI, you know, how does that all work?
Yeah, it would have to be an ancillary diagnostic. So before it would, and then we would move that into a clinical trial. So we do have some great mass spectrometrists that are actually in the surgical suite.
Mm.
For these preparations, they would be probably similar to what a pathologist would do, is take them out of the surgical suite and prepare them with the immunohistochemistry like workflows. The time to doing that is really dependent on the right conditions. And I think that we're really close to being able to do that. There's a lot more interest in doing this now. So maybe within the next 2 years, someone will make that breakthrough.
Great.
Yeah.
Okay, thank you. Next up, we will have Richard Drake, also from MUSC, who is going to talk to us about imaging glycans and intact FFPE tissue sections.
Thank you, Mike and Sean, for the invite. It's been an amazing meeting so far. Hope to continue that. Jeff and Peggy did a great job of setting up the method, so I may blow through a couple slides because you never, but I'll put it in context. And as Peggy mentioned, a lot of times we'll run the glycans and then pass the slide over because it just gets them out of the way for everything Peggy just showed you. So we can do data integration, pretty routinely. Very brief, we've spun a couple things out around this technology, especially on the glycan side. And so what we talking about on we were already in the ECM, so now we'll look at the glycocalyx part of the ECM. A very simplified version of that, but just from a contextual standpoint, this is where you're going to find your mucin glycoproteins. There's glycolipids out there, lots of surface glycoproteins, all the proteoglycans. And of course, that's what pathogens bind to in immune cells, viruses, receptors, et cetera, all the glycosylations going on the surface. There's nothing really simple about it. And so our attempts, we're kind of on the front end of, where you can go. So that's why the enzyme part's very critical, because at least there's some tools there. Most of the time it's reductive. We're just getting rid of something or chopping it down so we can detect it in the right mass range. And unfortunately, the Peggi for N-link glycans, it's been around a while, and it's really specific for these, is this a pointer? Well, the N-linked glycans right here. I don't know if one of my skills. So in humans, there's 7. We use these little cartoons to define these sugars that get combined in many. There's other ones in the other conjugates for N-linked. There's 7. If you're a mouse or not human, you get 8. You get a funky N-glycylneurominic acid version, a new GC. So, and we can pick those up pretty well too, but I don't think I've got any mouse studies today. As Peggy mentioned, it's been very fortunate to work with Peggy and our colleague on in Meta. Peggy has also a huge glyco interest. And so we all worked, developed a lot of technologies together, which I'll summarize here in a second. We're now on 11 years since we did the original imaging MS, and it has come a long way. And, I think as Jeff mentioned, once that timsTOF hit, that's just taking it to another level. Speed, accuracy, the image quality, it just lets us do so much more. And working together with Peggy on and now that we're all in one place, we've done a lot of spin-off methods. So it all started with the imaging on the tissue, and then we started doing ancillary experiments for everything else. Essentially, it's like you can get it on the slide. We got the method. The back end was already set up. So as long as we started with something with a glycan on it, we just applied the same method and put it in front of the timsTOF. Multi-imaging. Anyway, so now we're doing glycans. So these are released by the Peggi or whatever enzyme we put on there. And so we'll cover that. A note on the methods. We worked out the antigen retrieval. For whatever reason, citroconic acid, a little acidic, just works beautiful and fairly reproducible, but it's pretty flexible. And I'd say, and as Peggy indicated, we can do any IHC we want now once she worked that out. And then, there's lots of carbohydrate antigens. And we started going down that path too first, and then start linking antigen expression with the glycans themselves. And then there's a lot of different things, no time to cover. There's a lot of places you can do different things that they're either complementary or, just generate new types of data. I'll cover a couple of those briefly. Oh, there's the timsTOF. Anyway, some things we've noticed over time now. Well, it works on FFPE. That was kind of the whole point. So, right out of path. So we don't do anything, other than what's already been done from the surgeon to the pathologist to the embedding and the reading. So the other great part about the enzyme aspects, the signal's dependent on the enzyme. I mean, if the enzyme didn't work, we're not going to see anything. It's turned out to be very reproducible as sprayer technology is also improved significantly over the last 11 years. And with the glycans, now we can't even get into the isomers. That's a whole other thing. And we got a lot of tricks for that. But essentially, there's generally not more than a couple hundred. So that it's manageable, on the data side. And as I mentioned briefly, they're always the same. They kind of have to be. And so, again, isomers are different, but the masses are the same. And we really are now, I think, looking at a glyco code, which then, of course, in all this analysis we're talking about, that can be useful for integration. I won't spend time on this other than we've worked on a lot of different tissues, and all these are ongoing at some level, particularly the prostate and breast, and we're spinning out all kinds of colon, pancreas studies. And our colleague Anand has always been a liver guy. There's just some amazing stuff we've been doing in the liver. And then, as Peggy mentioned, working with Mike on different things and working with Sean on Alzheimer's for applying these methods to all human FFPE tissues. So this is my little show pony as far as, if you don't want to call it again, it's a nasty tissue. But if we were playing the pathology game, we already cheated. There's your blue villus adenoma, circled by the pathologist. The black wore off, so I had to put a different one in there. So it's a piece of colon. You can see the crypts turning into this adenoma that then turns into this tumor all in one slice. And it's just not subtle. The glycans have always been spatial, extremely spatial, and there's nothing random about where they're showing up. And like in this case, especially when you get a colon tumor, there's 174 individuals, glycan species. If you start layering in, there's 1,000 things on here because of fucose isomers. When you get branches, it's just endless. But the core structures, very robust, and they segment beautifully. And this one, I mean, that's the beauty of it. If you give me any tissue, human FFPE, I'm going to see these. Now, this is, of course, the abundances that will change and the specificities. And so these are very straightforward, but it's again, almost a QC. And what we're finding with disease, particularly cancer, but also on the diabetes, obesity side, you just start adding sugars. The whole sugar regulation pathway is messed up, and you just keep adding things. And they can get very complex. And I know some of these exceed the detection limits eventually. So it's pretty robust. And I'll mention every organ has its own glycome. Again, underlying it, there's a foundational part, but it has different components. We've done some clustering so far, especially on the normal. The thyroid stands out. If you walk it down, the kidney's the other one. They just don't look like other tissues. And then you throw in a tumor, the liver tumor really pops. And this is a kind of an atlasing project that we've got submitted as we go. So again, just kind of keep in mind, each tissue, each organ has its own glycome, just waiting to be, imaged in this case. So we've ended up with a platform. I call it a glycan hammer, and everything else is a nail, however you want to use these metaphors. But essentially, we can put just about any, if I can keep it on the slide, we can spray it with the enzyme. And then this back end is always the same. You have to, change your spatial, parameters, but it's essentially the output, and the type of data you generate, because it's so consistent, is also kind of baked in the cake already. We have sprayed a lot of different enzymes, and I didn't have time to go through. I mean, but it's essentially, if you have it, it isn't. Would love to talk if Stacy's back. Her mucinase, we've been working with her as Peggy developed for collagen assays. I spray the sticky enzyme at our site, and then I image one slide, send the other one to her at Yale. She does all the LCMS, and then we bring it back together, and that's about getting wrapped up for a paper soon. And we've overall developed essentially, especially a cancer patient, if they walk in the door, go through surgery, et cetera, treatment, any tissue that's obtained, any fluid that's obtained, we can get it into this platform now. And the not one spatial part, but you'll see where this is going. We've cracked the immune cell glycome recently and the ability to capture them. The initial method paper is already published. It's from Jake Dressman, who's a student in Anand's lab. But again, it's one of these things where the three of us all work together on different components. Interestingly, it was not the method, but the new laser source that we developed that the government's pretty interested in. So I don't know why. Here's the spatial part. Well, it's also special, of course. First one to do that, right? Spatial special. We can see the naive T cells, right? We can see the cells. They're captured on the spot. This is an antibody capturing CD4s or CD8s. And so there they are. Now you can start, I mean, we can generate beautiful signal, and now it's just a matter of resolution, instrumentation, optimization. You know, we're going to get there to be able to, you know, is this glycome different than this one? Probably not, but let's see. I mean, it's nobody's looked. And we just cracked, this is unpublished right now, and I'm just going to summarize the structure because it's going to come back here in a second. It isn't subtle, difference between CD4s, CD8s. If you start messing, if you stimulate a T cell, guess what? The glycome completely changes the reflective. We're just scratching the surface. CAR Ts, of course, I mean, just don't look like these PBMCs. And then the B cells are a whole other class. And so we're finding this is going to be a very fertile area for moving forward. I got two quick examples. One is, what about the immune cells in the tissues? You're probably sitting there going, gosh, I wish I knew that. Well, we'll show you where we're at with that. And then a new technology part that does kind of link in beautifully with proteomics, but ties right back into everything, targets. So didn't read the room right because I needed a positive example of an immune a very active immune system, something where we knew that it's just going to be active. A COVID autopsy lung is going to cover that. Should have done the TB granulomas that I work with Mike on because they're also beautiful in the same system, but that's a different, happy to talk offline. This, originally, my colleagues at the FDA just did a bunch of single IHCs to link where the CD8s and the 163, you know, the different markers, and we started lining up, and we can start mapping the individual glycomes co-localized because we could just co-localize everything to the same tissue. A lot of times we'd start with the tissue that was stained already, and then there was no challenge to line it up. So this was published already. But then I started looking at this prostate tissue is the one that struck me, and we kind of got off there's so many other things you could look at. The little immune clusters in most tissues we just weren't looking for anymore because most of the time we didn't see anything. And then this guy popped up, and it was, if we're ever going to see an immune cluster glycome, that's this guy. It's like, we did. And actually, again, we can start mapping it. Now, some of these glycans have other functions, and so, but if you underneath them is exactly where these immune clusters are. And there's a theme about bisecting and high mannose glycans, which we know now from PBMCs or B cells, in which I'm not showing it right now, but that's what most of these guys are from other methods in that cluster I just showed you. Briefly, I got permission from Albert to do the supersized colon polyp because there's like another name for it. This is roughly the scale. The thing, it's on a slide, but it is immense. And so it has a lot of things going on. The immune, and this is a like kind of more normal polyp to the left of it. There's some immune clusters in this guy. And so this is just 4 different glycans. There's a lot going on in this tissue, and anything colon is beautiful for N-glycans anyway. But we homed in on this region. You see where these immune clusters are, and we can light them right up. And then you can do this overlay, and that's great. So as Peggy mentioned, we've been working on some Amergen panels, specifically to kind of do some subtyping. Well, what's the immune cell subsets in these things? And they're lighting up quite nicely. And actually, Lindsay Young, who's back of the room, did this. And you can see how we can link, so that it is mainly B cell composition and some Ts. You can link the glycan with exactly, we can do this anywhere on this tissue, essentially, and get the proportions of what's present. And so, and briefly what we're seeing, these immune cells are we can see them. This is far a long way from single cell, that lone T cell doing something. Most of the tumor tissues we have, we don't see much, which kind of fits. It's an immune environment that's hostile. There's no nutrients there, or it's purposely being shut down. And so the glycans are reflecting it, and that's our hypothesis, that when we get these tissues, we're getting a snapshot of what it was going on at the time, even though it's a dead piece of tissue now and cross-linked. So we're trying to put a metabolism interpretation on N-glycans as a readout. And I can quickly go through my other example real quick. Sialic acids, always critical. Historically, on the path side, Sialyl-Lewis X and Sialyl-Lewis A. Sialyl-Lewis A is C19-9. Sialyl-Lewis X is you ever watched the T cell rolling movie? It's binding Sialyl-Lewis X on a selectin on the endothelial surface. So they're good players, bad players, but classically been associated with tumor antigens. So Vivian Liu, also now working at Stanford, and with Mike and Sean, back of the room, also developed a bioorthogonal chemical labeling probe using a sugar. Seems to be popular at Stanford, that kind of approach. Dr. Patozzi, of course. Anyway, it's yes. So, I mean, inspired by everything she's done with sugars and click labeling. And so the beauty of this is we can do the chemical labeling right on the tissue. You don't need a live cell, you don't anything, we just chemically modify it, and we get 2 things out of it. We get the isomers in this case, alpha-2, 3, and alpha-2, 6 with a shift, but we're also incorporating a click handle. You can do a lot with this now. This, of course, that was the beauty of the chemistry and why it won the Nobel Prize. You can attach things in that bioorthogonal fashion. So we're doing all kinds of things. And so with imaging, it's great because there's enough of a mass shift that it's not subtle. So we use it all the time just to do that. That T cell data I was showing you already incorporated 2-3 versus 2-6. So it just has all kinds of utility. But one of the things with it is that it has this click handle. So what if you put it as an affinity? And so we were doing different tissues where we characterize looking for 2-3 sialic acids as a prostate. And the beauty of this was also it's on the edge, so you don't need LCM. Like the tumor's right there. We can just scrape this right off. And we know where to look. We'd characterized it for, with the T for the tumor, where the different 2-3 linked sialic acids were. And then briefly this movie. So chemically modify, now we've got this handle. You can click on a biotin, or you can click it right to a bead. And so once you get that captured, now you can do all the proteomics you want. So we can get it linked. We can do usually we'll go trypsin first just to get what's bound to it. It's still a peptide attached with the sugar. So we come in with pangase eventually and do the LCMS on that. You get the peptide data. And this is a short version of kind of things. A lot of glycoproteins on this list. If you just scrape it off and run it, just what's there, there's 3,600 plus proteins there. we're getting this down to under 100, but we know these used to be 2 or 3 sialylated and also potential tumor antigens, and they were from the tumor. And we got this compared to the top part also. And if you're using antibodies, whether it's MALDI IHC or Mibitoff, well, this might be a new list. So we like this where you can just feed all this information back and start working with all these other approaches that we've been talking about to even just home in closer and closer and closer. So that's where I'll stop. Acknowledgements. The MAD lab here, as well as our colleague Lauren Baldwin on the proteomics side. And again, Helena Bai and Lindsay Young, postdocs who did a lot of the example work that I just showed. And yeah, this is for 2024 because we filled up. But so it's also after hurricane season, by the way. So intentional. And yeah, go glycans. So we'll stop there. Thank you.
Thank you, Richard. So we got several questions in the chat. If there's any in-person questions, please feel free to step up. Do you think you can apply your method on Sialyl-Lewis antigen-related diseases? For instance, leukocyte adhesion deficiency type 2.
Sure. If you got the sample.
What are your strategies in narrowing down to hundreds of proteins to analyze?
Mean on the after that step, or
Well, I mean, it's not my question. I don't know. Like,
sorry.
answer the question you wish you were asked, Richard.
Well, I think it was narrowing it down to hundreds was better than thousands. I mean, you know, so that was why I like that was the advantage of using that click chemistry, you know, to really target and enrich, you know, a known tumor antigen, something carrying a known tumor antigen. And a lot of, historically, we don't know most of the proteins that are carrying those. So it's kind of, yeah, so going from everything to a couple hundred was kind of the next step. You know, but I think then, right, you start narrowing down, well, where's a good antibody? And will it work beautifully with the MIBI, or will it work with MALDI IHC? And then you can start really getting down into it.
Have you seen examples where there's the protein is present in normal tissues and tumor tissues, but you see in tumor it's aberrantly glycosylated?
I know we have. I can't think of the example. I mean, even that prostate. So we're getting down kind of granular. So like prostatic acid phosphatase and PSA, which aren't great tumor markers in the tissue, but they weren't great markers anyway. But we are getting that kind of data after we do that Peggi step, because now we know which site they were being carried on, and we're starting to see that. Problem is we've only done it on a handful, and so it's like, you know, so it's like the tissue above it, which isn't tumor, is different than, you know, the tumor for those two types of glycoproteins. And so I'm sure we're going to see it, but it's will kind of come with the data, I think.
And I think your answer will be a yes to this too. But it is very cool how you profiled N-glycome on PBMCs. Do you think this can be applied on cytological samples?
Yes.
Okay.
If you can stick it on a slide, we can keep it there. That's, yeah.
And then there is this questions here about, excuse me, if you could expand a bit on stuff that you've seen with TLSs in tissue. Oh, tertiary lymphoid structure. So the lymphoid aggregates.
So we're just getting started, but like now I have a student working on this now on the prostate side, where we're cataloging about 40 different prostates And the one that I started with has like the most expansive list of glycans we can take. And it's a gradient down from 0. And then we're trying to figure out again, like location versus, so there's a general theme. The farther again, it's on the same slice, right? So it's not distal from the tumor, it's just not in the tumor. Those tend to light up meaning we can see a glycan signal better than the ones that are right next to the tumor or in the tumor. And with prostate, right, it can be a heterogeneous environment, compared to the other solid tumors. And so that seems to be the trend, though. The closer you are to the tumor or within the tumor, less signal. And I think it just fits with the, so this hostile tumor microenvironment or immune environment where, the deplete, and I think in pancreas, we're seeing some things where we're like, the energy is all in the tumor, right? It's just taking all the glucose and glutamine, oxygen, et cetera, and just being pulled into the tumor. I think all these clusters of cells that might be out there are just starving. And then, well, they're not making glycans. And, but we've seen that in different systems where, again, I'm trying to put a metabolic interpretation on a dead piece of tissue. So it's, we'll see.
Well, thank you, Richard.
Thank you.
So our last talk, it's a pre-recorded talk from a grad student in my lab, Leow, and she's gonna talk about how we've, in collaboration with Richard and Peggy, MUSC, how we've incorporated some of these glycan approaches into some of our high-dimensional IHC studies with MIBI.
Hi, my name is Kay, and I'm a graduate student in the Angelo lab. And today I'll be talking about some of my work using a multimodal MALDI imaging workflow to identify N-glycans that are associated with pro-invasive trophoblasts. Glycans refer to complex carbohydrates that are attached to proteins or lipids, and N-glycans in particular are attached to asparagine residues in proteins. And we know that more than 50% of proteins are N-glycosylated. And the biosynthesis pathway of N-glycans produces highly complex and diverse structures that could in turn regulate protein function and cell biology, such as immune cell recognition and cell migration. We also know that aberrant glycosylation in cancer cells have been observed for decades, and they've been implicated in hallmarks of cancer, including invasion, metastasis, and immune regulation. We're interested in studying invasion in the context of pregnancy as a highly organized and normal developmental process, and many interesting biology occurs at the interface between the maternal decidua and the fetal placenta during pregnancy. In particular, there are fetal cells known as extravillous trophoblasts, or EVTs, that are derived from the placental villi and could infiltrate into the decidua to facilitate the attachment of the placenta and remodel the maternal spiral arteries. They are also known to contribute to suppressing the maternal immune response against the fetal tissue. And we know that glycosylation has been suggested to be critical for placentation as well as regulating the maternal immune response. And it remains an open question how glycosylation could contribute to the invasion by extravillous trophoblasts. And we thought we could contribute using our knowledge of the spatial context of EVTs, knowing that each subtype of extravillous trophoblast occupies a distinct microenvironment. For example, the attaching trophoblasts at the tip of the placental villi, migrating through the decidua as interstitial trophoblasts, and gathering around the arteries as perivascular trophoblasts, and finally moving through the artery walls as endovascular EVTs. And we hypothesized that each of these subtypes could carry a distinct profile of N-glycans, and we would like to leverage on our imaging capabilities to understand the spatial profile of N-glycans in each of these microenvironments and therefore infer the role of glycosylation in this invasive process. So the key imaging technique that we use is multi-glycan imaging that was developed by Dr. Drake's group, where we use a de novo mass spec to visualize the spatial distribution of N-glycans based on their molecular mass. And so briefly highlighting several key components of this workflow, it's a highly robust method that is completely compatible with FFPE tissue, and the N-glycans are released using an enzyme, pingase F, that's highly specific for digesting N-glycans. And we use the timsTOF MALDI-2 to analyze the glycans that are released from the tissue section. And this gives us glycan images that are down to 10 micron spatial resolution. So in this study, to profile the glycans in the trophoblast subpopulations, we use a multimodal approach that combines the glycan information from MALDI with histology from immunofluorescence and H&E. And we perform imaging on simultaneously on the same slide to directly match histology to the glycan phenotypes. And we use a cohort of decidual samples that from the first and second trimester of elective terminations of normal pregnancy. We perform a 3-plex immunofluorescence to obtain high resolution images. Then we washed off the antibodies following steps that were recommended in a recent paper by Dr. Angelo's group, and directly applied the glycan imaging workflow on the same slide, then washing off the matrix for a H&E stain after MALDI. So this gives us images from immunofluorescence, H&E, and MALDI. And our image analysis comprises firstly annotating and masking the EVT populations using the H&E image and immunofluorescence image of HRAG, which is a marker for EVTs. And we manually mask the HRAG positive pixels around regions that are corresponding to the trophoblast histology, followed by co-registering the immunofluorescence mask over the glycan images. And so for each of the IF masks, we quantified the average glycan intensities. So in total, we annotated over 100 masks across the trophoblast subpopulations. And this is a heat map representing the average glycan intensities in each trophoblast subtypes, where the rows are the cell types and the columns are the glycans. And in total, we identified more than 100 glycans that were annotated by their structural features. And we'll talk in more details in the next few slides. But here we could start noticing glycan enrichment in each trophoblast population. Then we perform linear discriminant analysis, which is a supervised dimensionality reduction method, where we input the array of average glycan intensities in each mask, and then provided the model with the classifications of the trophoblast subtypes. And we plotted the LD1 and LD2 axis, which represents the greatest variation among the trophoblast classes. And each point here represents the glycan profile in each trophoblast mask. And we thought this was a cool result because this suggests that in LD1 on the x-axis is separating the glycosylation profile in the trophoblast in the order of the invasion trajectory from the attaching trophoblast to the interstitial trophoblast, followed by perivascular that are gathering around the spiral arteries, and finally endovascular trophoblasts that have invaded into the spiral arteries. Next, we perform differential expression analysis to identify glycans that are enriched in each trophoblast subtype, and differential expression was identified by significant p-value between at least one pairwise comparison. Here I wanted to highlight an example that we thought was really interesting, showing glycans that enriched in the attaching trophoblast at the invasive front from the placental villi into the decidua. And this could suggest a pro-invasive role for these glycans. And we noticed that they belong to a class of glycans that are characterized by branching, where each branch comprises a glucanate and a galactose residue. And when there are 4 or more of these branches, we call these highly branched glycans. And these highly branched glycans are interesting because we know that they're driven by an enzyme known as MGAT5 that has been associated with metastasis and upregulated in many cancers. And our preliminary analysis in transcriptomics suggested that MGAT5 expression was upregulated in the attaching trophoblasts, which would suggest that highly branched glycans and MGAT5 could have a pro-invasive role in placenta invasion. So in summary, we combine multi-glycan imaging with immunofluorescence and histology to determine the glycan expression in each trophoblast subpopulation to elucidate the roles of glycosylation in invasion. And we notice a progressive change in the glycan profile along the invasion trajectory of the extravillous trophoblasts. And we identified highly branched N-glycans as potential drivers of placenta invasion. So these are my acknowledgements. Please don't hesitate to email me if you have any questions or comments. Thank you.
Guys, who are you clapping for? She can't hear you. It's recorded. Just kidding. Anyway, thank you, Virtual K. And since she's not present, and since we're about 8 minutes over, why don't we go ahead and break for lunch, and then we'll have at 1 p.m. we'll have the talk from Aaron from Omero.